Welcome to OA Week 2025, everyone. We're going to be getting started in just a moment as we let more attendees enter the Zoom room. Welcome, everyone. Thank you for joining us for OA Week 2025, and actually for our final session of OA Week 2025. This is going to be closing us out for this year's OA Week. My name is Carly LaRoche, and I'm a member of the GOA-ON Secretariat. Please note that this session is being recorded. OA Week is being presented by 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the United States National Oceanic and Atmospheric Administration. Third, the IAEA OAICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And last but not least, the IOC, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization, UNESCO. GOA-ON was established in 2012 with just a handful of members. Since then, it has grown immensely to over 1,200 members from over 110 countries. GOA-ON also consists of 11 regional hubs, which span across continents and oceanographic regions. We've heard from most of these 11 hubs throughout OA Week. If you're not yet a member of GOA-ON, you can join today by visiting goaon.org. We'll drop that information into the chat in a little bit. OA Week debuted in 2020 and returned in 2021, when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022, OA Week has become an annual event to maintain momentum around OA research and to provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of OA topics and speakers from around the world. A few words on housekeeping before we get started. During the presentation, all participants are in listen-only mode. You are welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions, and we'll pose them to our speakers during the question and answer period, which will begin immediately after the final presentation. For the discussion, you can also use the raise hand function in the toolbox at the bottom of your screen, and we can call on you to ask you your question directly. And before we begin, I'd also like to express our sincere appreciation to Ian Ennox from NOAA's Atlantic Oceanographic and Meteorological Laboratory, who came up with the concept for this session and unfortunately wasn't able to join us today. But we have an amazing moderator who's going to be taking his place, and I'd like to introduce her for the session. Molly Phillips from the University of Southampton is a final year PhD student. Building a wide-range, high-frequency total alkalinity spectrophotometric sensor. I always mess that one up. And she's also one of our wonderful OA Week coordinators this year. So with that, I'd like to pass it over to Molly. Thank you.
Molly. Thank you very much as well to the audience for joining us today. This session is the Technology Development in Ocean Acidification Research session. So ocean acidification research faces numerous challenges stemming from high spatiotemporal variability, subtle long-term trends, and complex species responses. Addressing these issues requires high-precision measurement of multiple carbonate parameters across space and time, coupled with high-replication assessments of biological sensitivities. Technical, financial, and logistical hurdles remain, and we've actually heard a lot in questions this week all about those technical and financial difficulties from various community discussions. But emerging technologies that we're hoping to discuss today are helping to close those gaps. So yeah, this session will highlight advances in sensor technologies and autonomous platforms for large-scale monitoring, because together these innovations are accelerating our ability to understand and respond to ocean acidification. We'll get straight into the talks. So our first speaker today is Yui Takeshita. Yui is a scientist at the Monterey Bay Aquarium Research Institute. His work focuses on developing a sustained global biogeochemical monitoring network from the open ocean to the coastal ocean, utilizing profiling floats and underwater gliders. Yui, if you want to share your screen.
Thank you very much. Great. Screen's working for everyone? Okay, great, awesome. Thanks for the introduction. Thanks for inviting me to give this talk in the OA Week Technology session. My name's Yui. I'm a scientist at MBARI, like the introduction. And yeah, so today I want to talk to you all about the work that our lab and many, many other people have been working on to establish a global ocean acidification observing network using profiling floats and underwater gliders. I'll start with, acknowledgements. This, I'm really, presenting work on behalf of many, many, many people. This is an effort that's been going on for decades through hundreds of, researchers around the world and lots of funding agencies. And, I think it's a really exciting time because that, all the effort over the past couple of decades are kind of culminating right now into a global network. So too many people to acknowledge here, but yeah, I just want to acknowledge that it really takes a village or even a city to make something like this happen. So I think everyone is aware of the Argo network. So this is a snapshot of all the profiling floats of the Argo array around the world right now. So there's about 4,200. So each black dot is a float that has come up in the last 10 days. And so this, hugely successful program, and the core floats measure temperature and salinity. And then, but a subset of these floats now measure biogeochemical parameters. And then, so this is a part of the BGC or biogeochemical Argo network. And the 6 kind of approved, or the parameters are oxygen, nitrate, pH, chlorophyll A fluorescence, optical backscatter. So this is a proxy for phytoplankton biomass, and downwelling irradiance. And so, the goal of BGC Argo is to get an array of 1,000 profiling floats as part of the one Argo array carrying these biogeochemical sensors. And then for this talk, I'm going to focus on what we can do with an array of these pH sensors. So the pH sensors that are equipped on the profiling floats are the deep sea Durafet. You can see that here. I think many of you are aware of the Durafet. That's the pH sensor used in CFETs and CFOXs and stuff. But so it uses the same exact ISFET sensing technology, but it's packaged in a different way so that it can withstand high pressure. So it's the deep sea Durafet. And that's and this is integrated into the top end cap of a profiling float. And so a target array of 1,000 of these floats will look something like this. This is just a randomly distributed 1,000 ticks around the world. And where are we today? There's actually over 600 profiling floats equipped with at least 4 of these sensors around the world. The yellow dots are US floats, and the blue ones are international floats. And you can see that now, and the vast majority of the yellow floats have pH sensors on them. So you can see that we have really established a global array for the first time. So we actually have a global observing system of these key biogeochemical sensors. And to kind of give you, this is a fun GIF that I made. Ignore the colors, but the dots represent good pH profiles obtained from profiling floats from 2014 to 2025. And this is going to keep playing in the background, but this global expansion really has, it's really enabled by 2 major programs. The first one is both funded by the National Science Foundation. First one is the SOCOM project, started in 2014. So you can see this was funded to see the Southern Ocean. You can see the array really growing in the Southern Ocean first. And that's because of the SOCOM project. And so that started in 2014. Now it's in its 3rd iteration, SOCOM 3. But then the second program that's went in 2020 is the GOBGC, the Global Ocean Biogeochemistry Array. And this is research infrastructure program by NSF that was funded to deploy 500 profiling floats, BGC profiling floats around the world. So that is half of the target 1,000 float array. Historically, the US has done half of Argo. So this is really establishing the US contribution to the global biogeochemical Argo effort. And you can see around 2022, the program really kicked off, and now, starting from the Southern Ocean, now there are pH measurements around the world. And we have the longest record in the Southern Ocean because of SOCOM starting in 2014. And we put a tremendous amount of effort to make sure that the data quality is high from these floats. So this is a plot that's updated from Maurer et al. 2021. We try to take as many CTD-CAS discrete samples at the time of float deployment. And after we adjust the floats for sensor drift, we can compare the discrete samples to the float measurements. And we do not use the discrete samples to adjust the floats. So these are completely independent. And you can see great agreement between oxygen, nitrate, and pH is about 7 millipH standard deviation around the comparison. So we think, all these adjustments are made in real time, and so the data is science ready and publicly available to anyone who wants to download it from the Argo Data Assembly Center. So what kind of things we can do with the pH? So one thing that we think we can do, so plus 0.007, that's a little over double of the climate quality standards that the GOA-ON has kind of set as a target. But if you have a distributed network, and if this is a random error, we can still do ocean acidification without having all the measurements at the 0.003. And to demonstrate this, this is a plot of the Southern Ocean flows from 2014. And if you look at the surface, this is the surface 15 meters, I can't remember. You see there is a significant decrease in pH, surface pH of about 0.002, which is about what we would expect from the increasing atmospheric CO2. And this trend was, corrected or calculated using the best practices set out by Sutton et al. So that's the whole Southern Ocean from 30 south to 60 south. But now there's enough floats in different discrete, latitudinal bands. So if you just look at 50 to 55 south, you still get a very similar trend. So now we think that we can start to do spatial, ocean acidification trends, at least in the Southern Ocean, and hopefully in a couple more years around the globe. And so that's, I think that the data quality is high enough to actually directly detect OA trends. But what we can also do with these floats is take advantage of these empirical algorithms that are coming out. Lyre is a previous version, but now there's Esper, Canyon B. But these empirical algorithms are trained on the GLODAP shipboard measurements. And with the inputs of temperature, salinity, oxygen, location, depth, and date, you can predict TA, alkalinity, in the open ocean globally to about 6 micromole per kilogram. And if you combine that with the measured pH from the floats, you can drive other parameters. So DIC, saturation state, PCO2. So then we can, if you're looking at pteropods, and they're sensitive to saturation state, we can estimate that from these floats. And so I can give you an example of what we can do with these drive parameters, DIC. So this is work led by Guillaume Lineger at MBARI. He's a postdoc in Ken Johnson in my lab. So taking data from the Southern Ocean, again, because we have the longest record there, this is, this is a spatial map of the floats, the data. And then we can train a neural network, that combines both shipboard measurements and the floats, and then create a gridded data product of DIC. So this is DIC at 20 meters in August of 2015. So we can create a monthly gridded data product, one by one, gridded across the whole Southern Ocean. And he's done that from 2014 to 2025. And if you look at that, then we can actually see DIC accumulation across the whole Southern Ocean, but also spatial patterns. So now this plot shows the rate of increase of DIC in micromoles per kilogram per year across, various parts of the Southern Ocean. So parts, there are parts that are acidifying faster, taking up CO2 faster than other locations. And you can also do, transect views of the entire Southern Ocean, Atlantic, Indian, Pacific. So obviously the CO2 comes in at the surface, and then this is a deep water coming up from depth, so there's less, CO2 changes at depth. So the spatial patterns from this gridded product matches what we expect from the physical oceanography. And then you can finally create a trend like this. So this is from 2014 to 2024. So this is the DIC inventory. So the integral from the surface to 1,500 meters depth across the whole Southern Ocean for over the past decade. And so the black, so the red is the deseasonalized trend, and the black is the monthly integral. So you can see there's a seasonal cycle, productivity, and wintertime mixing, has the will change the inventory, but you can see the steady increase in DIC of about 0.92 petagrams of carbon per year. And this is in good agreement with the model-based estimates that the most recent recap program or project suggested. So I think we're in pretty good agreement with other methods. And so I think this is really exciting because now we have gridded product of DIC. You can make it for saturation state. This is just for the Southern Ocean, but I think we can make it global in a couple more years. It takes about 5 years for these trends to become significant. So, we can probably start doing this for the globe in a couple of years. And if you have a gridded product, then you can match the sparse biological data with these monthly gridded, estimates of saturation state, anthropogenic carbon, whatever you want, and then see if there's relationships with the impact of acidification on organismal to ecosystem impacts. I think this will I'm hoping that this will be really useful for people who are trying to look at more of the open ocean impacts of ocean acidification. But then Argo is really good at making measurements in the open ocean, but then the coastal ocean is a different story because they can only operate in 2,000 meters or deeper. So to kind of close that observational gap, we use underwater gliders. And so underwater gliders are kind of, they're platforms that can transect and profile to about 1,000 meters. And we have adapted the pH sensor, the deep sea Durafet, on the Spray underwater glider here. And this is kind of we published this a couple of years ago, but we just took the exact same pH sensor, made an adapter, and operated on a glider. So it's kind of a non-elegant way of doing it. It's hard to scale to a lot of different other platforms. But we showed that these pH sensors operated on spray gliders showed really good performance as well. So this is discrete pH on the x-axis, some shipboard measurements. Y-axis is the glider pH, and that's the one-to-one line. And the mean difference was about 3 millipH plus minus 0.011. And that's over 100 samples. And so we think that the pH measurements from the gliders are comparable quality as the floats. But again, so this was kind of a demonstration that we can do it on our gliders, but it's not really a scalable solution. So we developed a modular, we're calling it a glider FET, but it's really can be put on any autonomous platform. So we, shrunk the electronics, put it into a housing, and so that's a pH sensing element on the right here. So now this is, it's about 7-8 inches long. And so now it's a lot easier to be able to integrate into a wide variety of platforms. So it's kind of standard oceanographic, you turn on power, talk to it through serial, pull the data. And so it can be integrated into, we've integrated into AUVs, we've done it on wave gliders. So it's pretty straightforward to put on a variety of platforms now. And so this is a picture of that glider FET here and the wet bay of the next generation Spray 2 underwater glider developed at Scripps. As you can see here, it's in the pump sourcing. This is, just a prototype. We're not deploying it with tape for real. But so we've successfully integrated it, and so our approach right now is that we are trying to work with established observing networks for gliders and put pH sensors on them so that we can export this technology efficiently. And so, so there on the West Coast, there is a California Underwater Glider Network run by Scripps Institution of Oceanography. There's 3 lines here that's been sustained since 2007, I believe, and 2 new lines that were added a couple of years ago. This line up here is maintained by Oregon State. And on the East Coast, Robert Todd at WHOI maintains the Gulfstream network. So these gliders observe, they deployed in Miami, they go with the Gulfstream and are, recovered in Woods Hole. And these gliders currently measure temperature, salinity, oxygen, chlorophyll, and ADCP data. And now we have funding to integrate pH sensors onto all of these gliders. And we have made pretty good progress on that, I think. Currently, this is the map of all of the gliders within, our collaborations that have been deployed. So this is an active mission right now. There's a glider out there measuring pH in Southern California. These are our gliders, and this was the test mission from the WHOI glider network deployed at North Carolina because it was the first voyage, so we didn't want to do the full deployment from Miami. But the first full Gulfstream mission is planned in November, so in a couple of weeks. So we hopefully in a couple of months we'll have a measurement of pH all on the Gulfstream from Miami all the way to Cape Cod. So I expect that this would be, the number of these pH gliders will continue to increase over the next couple of years. So they're very exciting. So I kind of want to say, so if we have all those glider measurements, what can we do? And so the coastal ocean, as we all know, is very dynamic. And so we're working with the UC Santa Cruz Ocean Modeling Group right now to assimilate these pH and derived parameters, DIC alkalinity, into their ROMS model of the California current system. So they had a hindcast simulation in 2019. They assimilated this one transect here in February or April, I can't remember. And so and this is the full model domain. So this transect plot here, this is the difference between the glider observations and the model in their control run. So this is kind of their standard run where they don't assimilate any carbonate data. And you can see that there's large, so this is Monterey Bay here near shore. You can see there's large discrepancy of 0.2, 0.3 pH near shore. And this is problematic because this is kind of the source water for upwelling. So if we are off by 0.2, 0.3 in the models for the source water for upwelling, we are going to vastly underestimate the impacts of ocean acidification if we only use the model output. But not surprisingly, if you assimilate the glider data, then the model and the observations are in good agreement. You can see here. But what was also really interesting was that by assimilating one transect here, then it has a large spatial impact. So now this map on the left is showing the difference between the reference model or the two model runs, one assimilating pH and one without assimilating pH, and the difference of the average surface pH over a year. So you can see by assimilating pH in one location, you can have impacts of pH by up to 0.1, a couple hundred kilometers away. And that's because the ocean's all connected, right? So you change one property at one place and then the water affects, and you're going to know that impact. It has a long memory. So this, so we are, there's a paper actually in review right now, kind of summarizing the results of this experiment. But we do think that these observations are going to be really helpful in constraining and improving the accuracy of these models. And then, so now once we have these models that can, that basically interpolates all between these observations, then we'll have a more accurate data product. Again, then we can match with the sparse biological data sets to further understand the impacts of ocean acidification. How much more time do I have?
You have 3 minutes.
3 minutes. Okay, cool. So I will finish up with a couple other of the tech development, because I guess this is a tech development part session, so I should talk about that. So there's a couple of things that we're working on in our lab. So the first one, I think, so the Durafet, the Honeywell decided to discontinue Durafets a couple of years ago, and that kind of was, for us, it was all hands on deck emergency situation. Since it's been resolved, Honeywell has restarted production of its ISFETs, but we really realized that we are in a sole source vulnerability as the entire community. And then so we are looking into other ISFETs now. So we are working with a company, Lyonix International. The chip itself is shown on the right here. It is significantly bigger. So they will make custom ISFETs. So you can order whatever you want, which is great. And so this is a significantly bigger ISFET than the Durafet. And then so it's the Durafet ISFET is a 2 by 3 millimeter. This is 7 by 9. So it's an order of magnitude larger surface area. And what that allows us to do is put bigger O-rings on it and make the sensors more robust. So that is one of the big advantages of working with being able to make our custom chips. The performance of these chips are excellent. This is a comparison of pH on the x-axis and the sensor response on the y-axis. And each dot here is a spectrophotometric pH measurement. So this is basically testing the Nernst response of this ISFET. And we know the Durafets had a Nernst response. And this one, between the active range of the M-cresol purple dye, it seems to have perfect Nernst response, which is what we had hoped and expected, but we have verified that for maybe 5 or 6 chips in our laboratory. We are also making a design that's pressure compensated. So you put oil in the backside, which reduces the pressure coefficient. So it's a bigger chip, so it flexes a little bit more when under pressure, and that led to a significantly higher pressure coefficient than we wanted. So we have designed a pressure coefficient, a pressure compensated design here. So this tubing here contains the oil, and then as you go to pressure, it squeezes the tubing, and then the internal pressure of the sensor is the same as the external pressure. So it's pressure compensated. We've tested this on our glider in Monterey Bay. These are profiles of over a week or so using, it's not the fully pressure compensated design, it was an earlier version, but it seemed to work really well. This is the plot on the right shows the difference between the measured pH and an algorithm pH. And just from experience, we know that these algorithms do a really good job recreating pH under about 300 meters in Monterey Bay. So you can see great agreement between these two from the sensor. So I think this chip is looking very promising, and we just ordered a version 2. So we're hoping to start making more in a couple more months. And finally, I think many of you are aware of the dual pH alkalinity sensor that Ellen Briggs has been developing for a long time. And so we have started working with her this year. And so our role in our collaboration is kind of work on the high pressure side characterization of the chips because we are set up to do that. And so we're going to start work on that again in a couple months when these chips arrive. And so, so hopefully we'll have a better, we'll be able to characterize the pressure response and be able to deploy them on profiling floats and gliders. So with that, I am out of time. So thank you very much for your.
Thanks so much. You covered a lot of information there within 20 minutes. That was very impressive to have gone from the open ocean to the coastal ocean and then technology. I know that you have to head off now, so thanks for your time today. If anyone does have any questions for you, you can put them in the Q&A box, and then we'll pass them on to him once this session has finished.
I'll have like 5 minutes to look for Q&A. So, but thank you very much.
Absolutely. So our next speaker today is Emily Hammermeister from the University of Southampton. Emily grew up in Washington State, where she completed her undergrad at the University of Washington. She is currently finishing her PhD, she's now submitted, in oceanography at the National Oceanography Centre, Southampton. And her work has focused on advancing climate-critical ocean carbon observations through new applications of autonomous technologies. Emily, if you want to share your screen.
Yes, I'm doing it now. Can you see it? Awesome. And can you hear me okay?
Yep, sounds good.
Perfect. Thank you so much for that nice introduction. Yes, I just have submitted my thesis, so I'm kind of in that weird position of I'm a final year PhD, I guess. But today I'm going to be presenting one of my chapters, my second chapter, that is actually a really great segue from Yui's talk. We put some pH sensors on a really long distance deployment of a vehicle and tested how well they could kind of self-correct each other, as well as testing them against established models and algorithms. So yeah, let's get into it. I know this whole week you've already probably heard all of this fun stuff, but I'm just going to do a quick overview of the carbonate system. We know that it plays the marine carbonate system does play a very key role in the Earth's climate. And we know as CO2 dissolves into the ocean, it reacts with the seawater. It it forms carbonic acid, then that further dissociates into bicarbonate and carbonate ions. This releases hydrogen ions, which protonate the water and can affect the seawater's acidity, hence why we are all here. But there are other components to the marine carbonate system. There is DIC, which is the sum of all concentration of dissolved inorganic carbon, and total alkalinity, or TA, which is related to the charge balance in seawater. But Allison, I believe, will be getting into that a little bit more later. Both TA and DIC are measurable in our total quantities, so they are independent of temperature and pressure, whereas pH is temperature and pressure and is affected by temperature and pressure. And then the final measurable parameter of the marine carbonate system is the partial pressure of CO2, or PCO2. PCO2 is a measure of the concentration of carbon dioxide gas dissolved in seawater and can tell us a lot about CO2 fluxes at the air-sea interface and how much CO2 may be going in or out of the ocean to then obviously react and acidify the seawater. And I'll get into that a little bit later as well, because we looked at that in this work. So the final thing about that is it's important to note that, you know, in the marine carbonate system, between these four parameters that I just listed, you can actually fully constrain the system with any two of them. So you can use any two to calculate the remaining two. Anyways, again, I'll skip the gory details, but we are very aware that as CO2 has been increasing in our atmosphere, it's also been increasing in our ocean and therefore acidifying our seawater. And you can see in the plot, on the right, the Jerem plot, I don't know how to say that, but you can see that even the slightest decrease in pH can disrupt the carbonate system and result in kind of a major decrease of carbonate availability in the ocean. So by better understanding the dynamics of the marine carbonate system, we can better interpret, predict how ocean chemistry and marine ecosystems in general will continue to respond to the changing atmosphere and ocean acidification. However, the scales needed to kind of constrain all these parameters of the ocean carbonate system and processes and monitor ocean acidification have continued to pose a significant challenge. Higher resolution data in larger spatial and temporal ranges, as you can see kind of in this figure, are required for better ocean observation to provide accurate and usable carbonate characterization data. Luckily, just as Yui was saying, advances in autonomy have begun to offer newer, more efficient methods for data collection. Like you can see in this table, which shows some of the commercially available technology. So that's excluding a lot of stuff that's currently in the works, including some of the work that you're going to see in this session by Allison, what Yui showed, and some of even what I'm showing. And yeah, so you can see in this table, though, from commercially available stuff, that we still have a lot of room for improvement to try to fill some of those observational gaps, capability gaps. And so that brings us then to today. I wanted to talk to you about a deployment that we did to test the capabilities of these technologies by co-deploying two different pH sensors on an autonomous vehicle for a long distance kind of mission. So hopefully you can see this super fun animation, but this deployment was coined the Long Distance Proving Trial, or LDPT, because it was the first time that the AutoSub Long Range, which is the NOC's National Oceanography Center's kind of main vehicle, a lot of people might know it as Boatface, but we say ALR for short. It was deployed and recovered from the shore and had absolutely no ship support. So it lasted about 35 days. It covered 2,000 kilometers out and back, which you can see out is the kind of the black track of the ALR and back is the red dashed line. And it reached depths up to 1,000 meters once it did go out. You can see right now, once it did go off of that shelf, but it spent most of its time kind of in the coastal region of the Celtic Sea off the south coast of England. And it stayed pretty much above 150 meters most of its deployment, except for when it was able to go off that shelf. So this was a more coastal shallow deployment. The technical details of this deployment have also been published by Alex Phillips. You can see if you scan that QR code in the corner there. And again, there were two sensors on here, and we had a total of 55,000 total pH measurements in the total of this deployment. So of those two pH sensors we had on board the ALR, the first was the lab-on-chip pH sensor, which determines pH photometrically using metacressyl purple, or MCP, as the indicator dye, and it's a microfluidic platform. Again, Allison will talk a lot more about the lab-on-chip technology, so I will leave it up to her and not be too redundant. And the sensor is highly accurate with an uncertainty of 0.01, but measuring only once per hour on this deployment. The second pH sensor on board was Seabird's deep CFET, which Yui talked about a bit. It electrochemically determines pH using ISFET technology, and it measured every 1 minute. So it's a lot more frequent than the lab-on-chip sensor, with a slightly higher uncertainty of 0.0. So this is kind of what that setup looked like. We had both sensors in the front bay of the ALR, alongside a CTD sensor as well. And what's also key here to note is that they were sampling from an intake that was just about 40 centimeters below the waterline when the vehicle was at the surface. So the way that this vehicle was piloted, when it was at the surface, just bobbing there and maybe still receiving instructions of what to do for its next dive or mission, both of these sensors were still able to take water from the surface and get surface measurements, which is really key, especially for flux calculations, which we get into a little bit later. So the objectives of this work were really to see how well we could see, validate each other's sensors by having two sensors on board and maybe use them to correct, compared to established correction approaches using models or algorithms. And we wanted to see how down the line with other carbonate calculations that those pH corrections that we did, affected what we can calculate and how well we can constrain the system and kind of look at carbon cycling in this region. Because again, it's a coastal dynamic and shallow deployment. So what we are going to be referring to as the KO correction method is a method applied to the CFET sensor. So again, kind of how we saw a little earlier in the first talk today, this is because the CFET has a electrochemical reference electrode, and it's directly exposed to the seawater and sets the reference potential. That's just kind of a little bit of how ISFETs work, but I won't get too much into it. But that reference potential is known as the KO, and that's can be prone to drift or maybe a conditioning or settling period during its deployment. So rather than correcting the final pH, the sensor's KO is adjusted. And this is based on established methods used widely in the BGC Argo array. Basically, it assesses the changes in KO by comparing in situ pH values measured from the sensor against a reference pH, usually calculated from an algorithm or a model at depth. So I believe Argo typically uses around 1,500 meters for that kind of reference depth, because that's where you have a very stable and well constrained by the model's pH. So that's kind of what the pH reference that they use. And typically it's calculated based on, like I said, models. But for this work, we used the empirical seawater property estimation routine, which is ESPER. And we did that from locally interpolated regression, which is the ESPER-LIR, and then from neural networks, which is the ESPER-NN, which is based off the Canyon-B algorithms that you've probably heard of. And so we kind of wanted to test again, because like Argo uses these for those deep applications, that's where the stable pHs, this deployment did not get to those depths, at least not regularly. And so we wanted to see how well could this work in kind of more of a shallow setting. And instead of using those models, could our very highly accurate, stable lab-on-chip sensor actually operate as a reference? And so to kind of see our experimental setup, this is a table from the paper. We looked at the pH from the CFET uncorrected, so that's kind of in yellow, pH 1. We then used the lab-on-chip to correct the CFET, that's in green, and that was using the KO method. We just used lab-on-chip as the reference. And then we used the neural network, the ESPER neural network, that one's in kind of the pinkish red on the bottom, pH 3 there, to correct the CFET, to see again how well we could correct the CFET and see how well we could constrain the system. And then the other column that you see there, the TA input, we used algorithms for TA from both those same models, as well as just some basic linear relationships from Takahashi and Lee to use as a TA so that we could constrain the carbonate system with our pH input. So to get a look at some of this data, what you're looking at here is just the pH from the CFET and the pH from the lab on chip. There was no corrections yet. And so the bottom of those two plots is the difference. And you can see that that first week, I don't know if you can see my mouse here, but basically that first week is kind of that conditioning period. You can see that they didn't agree as well. And it slopes down to have a really good agreement, actually, considering no correction methods were applied. And that gray shaded area is actually their combined uncertainty. So they're sitting really well within their combined uncertainty between the pH and the CFET over the entire deployment. But there's still a bit of an offset, right? The mean is still 0.013. And you can see in the top graph that it's not a perfect overlay. Once we use the lab on chip to correct the CFET with that KO method, it becomes very obviously nearly perfect because we used it to correct. But most importantly, it's not just a single offset kind of like correction. This correction method works really well because it takes it looks at the standard change between the two and applies the correction, which is why you can see the conditioning in the settling period there at the beginning was corrected for really well. On the other hand, when we compare just the CFET to the neural network, so the modeled pH, there's a lot bigger of a difference than what we saw with the lab on chip. There's a lot more variability to begin with, and then also just a bigger difference between the two. And you can see again with that gray shaded region on the bottom plot that the difference between the two was kind of way out their uncertainty range at times, or the combined uncertainty range at times. When we used the neural network to correct the CFET pH, obviously we got that mean down quite a bit, but that standard deviation still stayed pretty high. There's a lot of variability there. You can see, especially kind of in the latter half of the deployment, this simply just didn't do as good of a job as the lab on chip. But I will note that it still was able to correct that initial first period of conditioning really well. So this is kind of just a high-tech version of, or a look at KO adjustment in action. And so the first one on the left, the lab on chip KO adjustment, you can see there was not a lot of work that needed to go into correct. Those segments are basically the breaks when the rate of change between the two, without getting too technical here, between the two were greater than the threshold that was set, which in this case was 0.005, then you'd have a breakpoint and you'd get a new rate of change. And that's kind of the way that the method works. On the right, you can see that we had to even set the threshold even like way higher than the lab on chip. We set the threshold to 0.1 just so that we wouldn't have as many breakpoints. But you can still see we had a lot of those breakpoints throughout the entire deployment, and we had to do a lot of those different adjustments, which is those red lines, those rate of changes. I will still note though, again, because the lab on chip was so less frequent than the CFET, there's obviously a lot more, less sample points on the left than on the right, which you can see by the x-axis of each one. And so this is kind of just showing a general distribution of the differences between the lab on chip and the CFET uncorrected and the CFET uncorrected with all the other ones, and then the corrections, which are here. The main takeaway from this plot, without trying to spend too much time looking at it, is that the pH sensors agreed better with each other than either sensors agreed with the models before correction. After correction, obviously the correction methods work, but you can see by the error bars there that, again, we had a lot more variability when we used the neural network to correct the CFET pH versus when we used the lab on chip to correct the CFET pH. So moving on to some other calculations, the PCO, we wanted to see what we could get from this data. So we, like I kind of showed earlier, we used those different TAs to test out how we could calculate PCO2 and what we got from that. And so this graph is showing basically each distribution of PCO2s we got, with those dashed lines being kind of the standard deviations and variability, and those red dots being outliers, basically. And again, the main takeaway from here is that the TA did not have a big difference. No matter which TA we used to calculate PCO2, it did not make a big difference at all on the final PCO2. What did make a big difference was which pH we used as the input parameter. So basically, when pH was held constant and we changed TAs, the PCO2 only varied by a maximum of like 3 micromoles or 3 microatmospheres. Whereas when we held TA constant and the pH changed, the PCO2 changed by up to 42 microatmospheres. So it was a lot different. And you can see again on the far right, when we used the neural network to correct the pH and then further down the line calculate the PCO2, we did get a lot of outliers and variability. And that's kind of a nod to propagated error as well. When you look at the same thing as a time series, and this is now only like the only differences here are by pH. So as we saw, the TA didn't make that big of a difference. So instead of showing all 12 lines on here, it's just by pH. You can see that there's a systematic, difference between the final PCO2 we're getting based solely off of the correction method that we used on our pH. And that is amplified by, the depth that we are measuring at. You can see this is the track of the ALR. And so, yeah, that was really telling us, okay, this is important, what pH we're using, how we're correcting it for further products down the line that go into, a lot of decision-making and global budgets. So on that same note, we were able, because we could get PCO2, we were able to also calculate the air CCO2 flux from these observations and algorithms. And again, you see that systematic kind of difference between flux calculation based on which pH correction method we used. And the green is the one that where we used lab-on-chip to correct the CFET, which is why it's a little bit bolder there. Because as I showed you earlier with the correction methods, we trusted that one the most. And again, what's a really, key point of this is based on the correction method that we used with pH, it actually changed the direction of flux. So this corner here had really high winds, and when we were using the neural network as the correction method, it actually showed us that, there was a positive air CO2 flux, meaning it was outgassing instead of absorbing CO2. And so had we used that the whole time without even looking at the other correction methods, we might not have ever known the actual direction of flux. So I think stuff like, this is really important to look at. Okay, so I don't know if I have no idea where I'm at on time. I think I might have zoomed through that, but that's okay.
You've got 2 minutes.
Some of the oh, really?
Yeah.
Oh, okay. So some of the key takeaways here, other than it being, this was a very new deployment it was a new deployment for the vehicle itself. It had never done this kind of shore to shore, deployed from Plymouth, recovered from Plymouth, the beaches of Plymouth. It had never done that before, let alone with pH sensors on board. So this was all around new for everyone. But also we learned that in these kind of settings where we're way above these deep, stable waters, models do underperform in coastal settings for monitoring pH. So at the end of the day, two sensors is better than one always, and it's better than a model in this deployment. As well as calculated PCO2 and CO2 fluxes, we learned were really sensitive to pH input, whereas they just didn't, they were a lot less sensitive to TA input. We learned that the Celtic Sea during this time is primarily a CO2 sink, and that small pH differences, like I just showed, can actually reverse the CO2 flux direction. So a lot of this is really a nod to what kind of pH sensors are we using, how are we monitoring them, how are we correcting them, and how can we use that to monitor ocean acidification and make sure that we are doing it as accurately as possible. As a self-plug, this is a publication in preprint now, in review, but here's the preprint. So if you do want to, there's a lot more to this paper. I really just kind of skimmed the surface. So if you want to take a look at that or reach out to me at any point, I really like this chapter. I love talking about it. So feel free to reach out. And I don't know if anyone's even on, but a huge thank you to all of the co-authors. They were really instrumental in this work. So with that, thank you. I think I hit my time okay. I think we'll do questions later as well, but I appreciate the invite to speak today, and thank you for everyone who is in attendance and listening.
Thank you so much, Emily. Yes, so if you do have any questions for Emily, you can type them into the Q&A box now and Emily can answer them throughout. But Emily's left all of her emails and stuff here if you have any questions at any point later. We will now move on to our next speaker. Let me see if I can share the screen. Our next speaker is Alison Sharp. Alison is the Associate Head of the Ocean Technology and Engineering Group at the National Oceanography Centre in Southampton. She is a research engineer who focuses on engineering devices at the microscale to create new technology for environmental sensing. She is also involved in the deployment of these devices, and we should be hearing about some of that today. So Alison, if you're ready to share your screen.
Absolutely. Thank you, Molly, for the introduction. Can you give me a thumbs up if that's showing all right? Looks good from here. Is that a thumbs up? Okay, there's a sideways thumb. That was very non-committal. Thank you. All right, so hello, everybody, and welcome to the latter half of the session now. I'm going to be presenting on the development and some example applications of a new sensor for total alkalinity, which can operate autonomously at the sea. And while I'm presenting, obviously, this is the result of work from many of my colleagues, primarily from the Ocean Technology and Engineering Group here at the NOC. I suspect in this session, I don't have to anymore say too much about why we want to measure alkalinity, because the previous speakers have addressed this quite nicely. But just to summarize, this is one of the several parameters that define the marine carbonate system. And in order to measure physical and biogeochemical processes, including ocean acidification, we need to characterize or measure at least 2 of these parameters, ideally more, especially if you're using autonomous sensors. Like Emily said, 2 is better than 1 and 3 is better than 2. In particular with alkalinity, if you're trying to study the growth or health of ecosystems that have a lot of calcification type processes, so this would be environments like coral reefs, things with shells, so mussels, oysters, coccolithophores, that sort of thing. Alkalinity in particular is quite an important thing to measure because the growth of those shells uses up alkalinity from the seawater. However, there are very few in situ sensors that exist for automated analysis, especially if you want to use them down deep. We saw an excellent example from UE at the start of this session of one that's based on electrochemistry, and this is using a very different technique, which has different strengths and weaknesses to that kind of technology. Specifically, the method that we're using, which I'll show you a bit before showing you this technology. For those of you who are familiar with alkalinity measurements, it's the single point open cell method. What that means in practice is that we take a pre-prepared solution, which I'm referring to here as titrant. So this is hydrochloric acid and a pH indicator. We pull into the instrument a fixed volume of that and mix it together with a fixed volume of seawater that's also been pumped into the instrument. After these 2 mix together, they get pushed through a gas permeable membrane, or pushed through a gas permeable tube formed by, pushed through a tube formed by a gas permeable membrane. There we go. On the other side of the membrane is some sodium hydroxide, which has a high affinity for carbon dioxide gas, which is created by this mixture. That gets removed, which we need to do in order to do a good measurement of alkalinity. So after the CO2 gas has been removed, this mixed solution with this pH indicator goes into an optical cell, which can read the color of the solution with the indicator in seawater. So if you go through some calculations, you can basically calculate the pH of the solution, having now acidified it, and that can allow you to calculate the alkalinity of the seawater at the start of this process. On top of this sort of measurement technique, we also do some lab calibrations of the instrument to account for its sensitivity to temperature and salinity, to account for the exact properties of the optics that are inside the machine. And in the field, we also calibrate it against reference materials, which are carried on board the instrument. So what that actually looks like inside, you can see some of the inner guts here. The main device in the middle there is what the instrument looks like. The black round thing at the bottom is a microfluidic chip. So this is a small piece of plastic that has very small channels cut into it, that kind of the size of a human hair. And that's where all of the chemistry is happening. So they're very small, which allows us to use very low volumes in order to do these chemical analyses. Material, seawater, or chemical reagents are pumped in and out of those channels using a syringe pump. So this is a pump that works a lot like a syringe in the lab. You can move something up and apply suction and push it down to create overpressure and pump things into channels. All of this is controlled by some pressure tolerant electronics, which are pictured through these pictures. These are pressure tolerant because we don't want to have to use a pressure housing for this device. Instead, we use just a plastic housing that has oil inside of it, so it's pressure compensating. And all of the pressure, all of the electronics inside, as well as the pumps and the valves and everything, are experiencing the same pressure as the ambient environment around it, which could be up to 600 bar if it's at the bottom of the sea. Last but not least, we are measuring all of the results of this chemical measurement with some inlaid optics that are inside the device. What this looks like in real life, I've actually got one here. It's the advantage of doing a talk from your desk instead of a conference. You can see it's about the size of my head. It's got all that stuff inside of it that I just described. And in order to put it on something that doesn't have a shell, so for example, if it's used on a mooring or on a lander, we would put this optional housing on top that's labeled in the picture on the right, where we would have flexible bags that contain the titrant and the sodium hydroxide, so these chemical reagents, as well as some reference materials with a known alkalinity. The sensor is on the bottom in that picture, and the seawater is pulled in through a syringe filter pictured there, the little green circle. And that just stops any particles from coming into the instrument. This is useful for making sure we don't block our tiny little channels, but also to make sure that no particles are interfering with the optical measurements because they would scatter light. The whole instrument takes about 2 watts of power while it's running and has been rated and used in the field down to about 6,000 meters deep, although I think it can actually go a bit deeper. I've got some examples here of some lab data that we've collected, which we use to validate the performance of the instrument before taking it out into the field. In this particular example, there's 2 CRMs, certified reference materials, so the famous carbonate reference materials from Scripps, which we use to calibrate the instrument. So those are shown on this graph with the X's. And then we mix different aliquots of these, so different ratios of the high and the low alkalinity material, and those are measured by the sensor to determine their alkalinity, and that's in blue. So the bottom axis is showing the expected alkalinity, and the measured is on the Y. Underneath, because the error bar is very small, I've just zoomed in and showed the error, so the offset between the expected value and the measured value. And as you can see, both of those have turned out quite well. The mean errors are very small. And in order to do climate measurements with alkalinity, our target is to stay better than 2 micromoles per kilogram in both precision and accuracy. So we were quite happy with this data. Of course, when you go to the field, nothing works quite as well as it does in the lab. So I've made this somewhat stupid choice of showing you my worst field data first, but it's also the easiest deployment, which is why I thought I'd start with this. So here at the National Oceanography Center, we have a pontoon basically out the back door, and we're at the junction of 2 rivers and the sea. So it's quite tidal. You can see the salinity graph in the top showing that the seawater is mixing with fresh water from the rivers. And those rivers also have the slightly unusual property that they have a lot of alkalinity in them. Unlike usual freshwater is often very low alkalinity, here it's twice as high as seawater in our rivers. So there in blue, you can see data from the TA sensor going up and down with the tides over the course of about a week and a half. And the black X's just show the results from analysis of some bottle samples, which we tried to take across tidal cycles in order to validate the performance of the instrument. So as I mentioned, you've got this inverse alkalinity-salinity relationship, which is plotted up in the top right for anyone who's interested. The mean error on this was not as good in the lab as it was in the lab samples that I showed in the previous slide. On average, we were offset by almost 6 micromoles per kilo with a lot of variability. I think part of the reason for this was that the tides change really, really quickly here sometimes. And during the period of the fastest tidal change, we actually saw changes of up to 5 micromoles per kilo per minute of alkalinity, which basically means if I grabbed my sample even a minute after the sensor finished its reading, I'd be off by quite a lot. So we didn't I don't think we were quite that precise in our time recording because we didn't expect it to be quite so extreme. We've also used this instrument in some slightly deeper environments, so we're going to be going deeper consecutively. This is a demonstration that I have from the North Sea. So we were at the seafloor at 120 meters deep, about 150 kilometers northeast of Scotland. And the sensor is on this lander. It's hard to see in the sea of gray tubes, but it's the one, if you can see my mouse, slightly to the right of center. It was powered by a battery and left down at the seafloor for 2 weeks in the context of a project trying to evaluate different techniques for looking at the detection of CO2 in a carbon capture and storage context. We did something a bit different on this deployment because it was one of the early ones and we wanted to do as much as we could for quality control, which is that we put an extra reference material on board the instrument and asked the instrument to treat it as an unknown sample and just routinely measure it a couple times a day. So in the graphs here on the right, you can see the salinity at the top doesn't do very much. The seawater alkalinity is shown in blue, and the alkalinity of this additional reference material is shown in the black X's. This is partly because we had a relatively limited amount of co-sampling that we were able to do in this environment due to the other activities happening. But this black reference material, these X's, gave us an average value of 2311 micromoles per kilo, and the certified value of the material is 2309. So we were really close, which is quite comforting given that the seawater was more variable and also the measurements from the ships that were in the area, there were a couple, ranged from 2301 to 2356. So there's a large range measured by the ships. And they couldn't actually get that close to the lander because they didn't want to smash into it with their sampling equipment. So this was having this extra material on board was a useful way for us to be able to independently do quality control of the sample analysis. This looks very similar to what the really cool experiment Emily just showed, although it's actually a rather shorter one. We've also deployed this sensor on these AutoSub long-range autonomous vehicles, the quote-unquote Buddy McBoatface submarines, for those who remember that name. You can see a picture of the submarine on the right there with its lid on. Rather more interesting looking on the left, you can see it without the lid on. You can fit about 10 of these lab-on-chip sensors if you so desire, if you pack them in sort of Tetris-like into the front and back of the vehicle. So we did this for a relatively short deployment in the Celtic Sea, so same area as where Emily presented data from in spring 2022 from one of the UK's research ships. The sensor was integrated into the vehicle electronically, so the vehicle could power the sensor and tell it to turn on or off. And the sensor transmitted its data back to the vehicle, which would then transmit it to the users over a satellite connection when it was back at the surface. The vehicle did a range of depths from fairly shallow down to just shy of 600 meters, and the ship sort of trailed it along doing some co-sampling sort of behind where the vehicle had recently been. I guess you can see there's these blue and yellow plastic things littering the front of the vehicle, and that's all of the reagent bags and reference materials for quite a number of different lab-on-chip, so this style of autonomous chemical sensors. So that was an example of where we don't bother using the top, but we just put these bags anywhere that we can fit them, basically. So here's a subset of the data that was collected on that mission. It's a little bit complicated because I've got both sort of a time series and depth variables, but in this case, I've just plotted it as a series of time, and the color of the dots indicate the depth at which the vehicle was traveling when it collected this data. So the dark blue at the start and end is fairly deep, so 589 meters deep as the vehicle is sort of going in the deeper end of the shelf. And then you can see on the other extreme, the 19-meter data in yellow. In addition, on this graph, there are these black X's, which are showing the alkalinity values measured from bottle samples collected as close as possible to these autonomous measurements. So there's about 33 hours of data, 144 sensor measurements, and the mean difference between a bottle sample and the nearest, both in space and time, autonomous sensor reading that we could get was 1.8 micromoles per kilo. Now, obviously, it's great to do things with an autonomous vehicle, But in this particular case, we could have gone with the ship and done some measurements. So the last example I wanted to show you was actually a more dynamic ecosystem where manual sampling and analysis would have been an absolute nightmare. So I think it's a nice example of why you might want to use autonomous sensors. So this is a coral reef tank. It's incredibly shallow because I think my sensor was about 3 inches deep. But anyway, you can see this picture. There's a CTD up at the very top, so measuring conductivity, temperature, and depth, such as it was. And an inlet for the TA sensor is this yellow circle. And this coral reef tank is actually here at the front reception of National Oceanography Center. So if you're ever visiting, you can come see it in real life if you like. This work was done together with a coral reef expert, Dr. Kay Davis, who's now at the Australian Institute of Marine Science. So thank you to Kay for a lot of expertise in helping to design this. So here's a snapshot of some of the data that we collected from this very alkalinity relevant environment. You can see these dark and light vertical stripes are representing day and night over the course of a few days. And we can see really clearly that over the day, so where there's sort of a light stripe, the coral reef is net calcifying. So the alkalinity is going down as the growth of the coral absorbs some of the chemicals that would contribute to alkalinity in the seawater around them. And then at night, we can see a small dissolution signal with an increase in alkalinity as some of the coral that was grown dissolves, and then it starts regrowing again in the next day. Particularly interesting here, which we were looking for, was the fact that you could see that there was a net calcification process. So every day it goes down, it goes back up at night, but it doesn't go up as high as it was before. So the corals are net growing more than they are dissolving. This is all going down. They then feed it, basically give the seawater extra alkalinity. But this was over a long weekend, so we got the chance to get a nice unperturbed signal before new materials were added to the tank. Obviously, in real life, we don't just want to measure tanks. We'd like to measure actual environmental ecosystems. This is something that Kay, who's pictured in that really amazing photo there, has gone and done in the Great Barrier Reef in Australia, and she's been working up the data. It's a lot harder in the real environment. You've got a lot more variables, but she's been able to prove that this technique works as well in other environments. So what's next for us? As I sort of alluded to, we're working up various studies and or planning other ones, looking at ecosystem analyses, looking at surface water CO2 uptake, and also at benthic carbon uptake or release processes. And of course, there's a very important application in contributing to the evaluation of marine carbon dioxide removal studies, especially ocean alkalinity enhancement. In the meantime, we're continuing to develop the technology. We've got a new version where we aim to decrease the volume of solution that is used per measurement. The one I've shown you today uses about 3.5 milliliters per measurement, which is small compared to lab tests, but if you're doing it autonomously, after 1,000 measurements, you started to make a fair bit of waste, which you have to hang on to within your instrument. And the new volume, or the new sensor version, is about a third of that. In parallel, we're also working on a new version, which we're going to combine with a dissolved inorganic carbon sensor, so that you can do both parameters on a single instrument, a bit smaller than the current one, which we hope will make it more suitable for use on gliders and floats. So that's it for me, other than to say thank you to my colleagues at the NOC who are listed there on the left, and also to our funders, which is primarily the EU and the UK's Natural Environment Research Council. And yeah, if you have any questions, do feel free to put them in the chat, and I've left my email address there as well, if you want to get in touch. Thank you.
Yeah, Alison. That was an enjoyable talk to not only see the technology, but also where it can be used. That's the thing that we've been asked a lot of questions about this week, is what technology can we use? What technology is there? So you're answering that question. Our final speaker, I will put up now. Our final speaker of OA Week not to put too much pressure on you, is Evan Lechner from the Woods Hole Oceanographic Institute. Evan is a research associate in the Department of Marine Chemistry and Geochemistry, working with Dr. Zhaohui Alec Wang, who was meant to be with us this evening. So thanks so much for taking on the session all by yourself, Evan. He is a marine chemist with a focus on seawater carbonate chemistry of coastal and estuarine systems. His current work focuses on the development and deployment of autonomous in situ DIC and pH sensors to explore spatial and temporal variability in the carbonate system across a variety of environmental settings. So if you want to share your screen.
Sure thing. Let's see. All right. Can you see and hear me well? We good? Cool. So thank you for that introduction. I'm really excited to be here. As Molly said, my name is Evan Lechner. I'm a research associate in Dr. Wang's lab here at Woods Hole. And I'm primarily responsible for the design, deployment, and operation of our carbonate chemistry sensors, sort of like at that crossroad where the lab meets the field. So I'm excited to talk to you about some of our work today. And I think the scope of the presentation is broadly going to cover so the initial instrument design of one of our instruments, this DIC and pH simultaneous sensor, from its initial conception through a couple of iterations to where it's at today. So there we go. I initially planned on doing a little bit of background in the carbonate chemistry, but I think both Emily and Allison did a great cover there, so I won't retread their work. But I will echo what Emily was talking about in terms of like there are a wide range of dynamic scales for the signals of variability you're looking for. So natural scales of tidal and seasonal variability are much larger than small scale ocean acidification or climate change related signals. So you have to resolve your measurements on a very fine scale in order to get at that kind of change. And similarly to what both Allison and Emily have said, your selection of CO2 parameters has a lot in terms of what error propagation can look like. And so to justify that, I've got this figure here from Orr et al. 2018, just sort of showing some of these kinds of reliable error propagation seen in carbonate constraining software like CO2 sys, just as an example. And so at the time of our instruments, like original conception, there were some pretty significant difficulties in the instruments that were available at the time, sort of like long measurement response time, long measurement cycles, which really made it incompatible for mobile platforms or deployments in like dynamic or extreme environments. So with that, I'll bring it to the channelized optical system, or CHANOS, which is the instrument we're talking about today. At its original ideation, the main goal was to do a 2 simultaneous parameter measurement so that you could constrain the carbonate system in situ. We ultimately decided to do pH and DIC. That's primarily what we operate with, but we've also developed the capacity to do PCO2 and DIC. The instrument is based on using a spectrophotometric technique for all of the parameter measurements, and we found that just sticking to that allows it to be a sort of a simple and modular design on the inside and the outside. And it also gives us the capacity to conduct higher frequency measurements needed for things like ROVs, profilers, and time series. And then lastly, we needed to incorporate a capability of doing in situ calibration using CRMs, much like what Allison described for our DIC measurements. So you can get a little bit of the idea of the internal sort of fluid components, and then you can imagine inside of this pressure housing is just a large set of electronic boards and circuits. So a super brief background on sort of the spectrophotometric concept is essentially relies on a pH sensitive indicator that dissociates between a nominal acid and base species. There's lots of different indicators that have a range of pH acid and base color spectrums. And so the indicator you choose will sort of give you a color change scheme that fits a specific pH range you're interested in. And what you can do is you can take the peak absorbance wavelength, so lambda-1 for acid, lambda-2 for base, and calculate the relative absorbances at those wavelengths, and back out what the initial pH was of your sample. It's a really convenient method for calculating pH because you don't really need to do any calibration. Your sample water is your own 0 point, and then you add the indicator to detect the change. So very simple and straightforward. You can then apply that technique to get to spectrophotometric DIC by rather than mixing your indicator directly into your sample water, you instead run it through a Teflon tubing length, similar to the gas membrane that Allison was describing. And in opposition to that, you pump an acidified seawater sample in a counterflow. And that allows you to equilibrate the CO2 of the acidified seawater across that gas permeable membrane and create a equilibrated indicator pH. And in the same way that you calculate change in indicator pH from your just general spectrophotometric pH, you can calculate a change in pH of that indicator and calculate its concentration of DIC with a couple of lab-determined constants. And so here you can see some figures from Mallory Ringham, a former PhD student in our lab, just sort of the conceptual design for, in particular, this equilibration tubing-in-tubing cell. So we've got like a spiraling concentric circle that allows us to fit about 2.7 meters of Teflon tubing into a relatively small, my head-sized brick of acrylic. So that's the core concept. The first iteration of Chanos, going back to 2013 and 2015, was a little bit more limited proof of concept design. It relied on syringe pumps to sort of back and forth pump reagents and sample seawater, which gave it a sort of a measurement cycle of about 15 minutes with 8 minutes of continuous measurements and then 7 minutes of recharging the reagents. And it was predominantly meant to be deployed on land-based estuarine environments, so it could be kept dry. And the sensor had an uncertainty and precision of 0.8 or 5.2 and 0.1 or 0.03. So near climate scale quality, but not quite there. And a lot of that has to do with sort of the same sorts of difficulties you've been hearing in some of these other talks. That the environment is different in the field than the lab, and coming over those hurdles with each deployment can be a little bit different. From there, the Chanos was brought into a sort of a 0.2 design, Chanos 2, under the supervision of another former PhD student, Mallory Ringham. And for this one, we wanted to get around that primary constraint of intermittent sampling and do fully continuous. So we installed a set of peristaltic pumps for continuous pumping of samples and reagents without any sort of recharge time necessary. We also prioritized a sort of more robust field housing so that it could be deployed in a wider range of environments. So to highlight just some of her work on this Pocasset River time series here in Cape Cod, we deployed it on a small tidal river nearby for about 59 days. What was pretty cool there was we were able to see a bunch of different episodic storms resolve themselves differently from sort of the background signal of the river. On the one hand, you can see this northeastern storm that was really typified by a dilution event or a precipitation event that diluted the typical seawater of the river there, which was very different from a tropical storm that blew through earlier on in the deployment, which was really dominated by strong winds and strong waves rather than precipitation. So you didn't see that same dilution, but you saw a lot of disruption of DIC-rich sediments and groundwater, which it gives you this spike in DIC. This system at this iteration only operated on DIC, not the pH parameter, but it had a similar climate scale quality of about 4 to 6 micromoles per kilogram. In the time since Mallory's work in 2024, we've continued to do more of these time series measurements on marshes. An example of this here, one that I worked on, was in support of a 2-year time series project looking at salt marsh drainage and lateral fluxes of carbon from the estuary out to the coastal environment. And here we're basically just doing contemporaneous continuous measurements 6 weeks at a time at a bunch of different salt marshes in the area. I, like Allison, have chosen to show you some of our messier data here. So The dots here are our hourly 12-hour tidal cycle time series. And you can see the fit of those to the DIC curve for the Chanos. And then these reddish orange are excursions of excluded data. That was, just not a good day, but that happens in the field. Since that time, we've also added a pH channel to operate on a similar continuous peristaltic pump design to allow us to do 2 measurements simultaneously. And so to highlight some of that work, we did a simultaneous profiling deployment based off of a float-based system in a salt pond here in Cape Cod. The float was designed to have 2 profiling winches, one for our Chanos instrument and another for an IFCB, which is a sensor that conducts flow imagery of suspended particles. And the goal of the project was to sort of look at biogeochemical changes related to the formation collapse cycles of harmful algal blooms. So it's a really fun environment for testing the instrument because it gets very significantly stratified over the spring and summer. So you get very pronounced chemical gradients over like an only 7-meter water column. And so as you can see in these graphs, which I'm not sure if they're zoomed in enough, we're seeing changes in DIC of approximately 400 micromoles per kilogram over that sort of like 5.5, 6-meter water column in just one cast, and a full point of pH in that same space, which is interesting and interesting to work with. It was also a great environment for just looking at the capabilities of our instrument. We had wanted to highlight that sort of higher frequency measurement and how to bring that out in the field. So we're capable of taking measurements about every 5 seconds for pH and about every 12 seconds for DIC. But then we also see a difference in the response time to change in sampled water. So pH is a more instantaneous response of about every 15 seconds, whereas DIC has a response time about every 5 minutes because of that long equilibration cell. And so what we did, knowing that, was we operated sort of a differential profiling approach where the downcast was sort of seamless, straight, and quick, focusing on our pH measurements that could respond more quickly. And then we used the IFCB tracking biological data, different sorts of dinoflagellates, diatoms, to pick prescribed depths to stall at for 5 to 6 minutes to allow our upcast to give full equilibration time for those DIC measurements, which was a fun way to look at that. So we've got some more stuff coming out for that project soon, but it's been a really interesting environment to work in. The last project I'll try to highlight for this instrument is the HabCam survey. So for those of you who are not familiar, HabCam is a Northeast Fisheries Science Center project, which is operated by NOAA, that looks to tow this vehicle you can see here and collect data about the scallop fishery. And so this vehicle is typically towed at about a speed of about 6 knots and kept about 1 to 2 meters above the seafloor. And so if you connect the ship speed here to those measurement frequencies I was just talking about, we're able to get a spatial resolution of about 15 meters for pH and about 36 meters for DIC. So it's relatively fine scale spatial resolution relative to things like gridded products for modeled data. Another really cool factor or feature of this project is that the HabCam vehicle itself is equipped with a suite of photographic sensors that are basically capturing bottom-focused photographs of the scallop habitat every 6 pictures per second. And then the crew takes shifts annotating those pictures for things like abundance, size, life stage, turbidity, and other unexpected animals. So we have this really neat data set to compare our chemical measurements with biological, ecological information. The vehicle also is equipped with a number of in situ water quality instruments for things like chlorophyll, oxygen, dissolved organic matter. So the goal for this project is largely to sort of look for connections between fisheries and ecosystem health of scallops, which is a really important economic asset in the Northeast here, and sort of how can we detect patterns between the chemical gradients and this biological data set. And so over the course of about a 1-week deployment, about 5 days at sea, We have about 1,300 kilometers of spatial coverage for pH and DIC measurements here. It's broken into basically 2 cruise tracks, one here off the coast of Cape Cod on what's called the Georges Bank, and the other one here sort of in between New York and New Jersey called the Mid-Atlantic Bight. And so it's been a really interesting place to work in. We can see some pretty clear spatial gradients, particularly over here in the Mid-Atlantic Bight, where we're seeing variations of about 0.15 pH and about 200 micromoles per kilogram. We think largely because there's sort of transitions between ubiquitous shelf water and deeper waters that are coming up the slope at the edge of the shelf there. Similarly, we can look at some of our other data and connect it to sort of identify different kinds of end member mixing in this space, sort of masses of water that are coastal, warm, and salty, typified of these Georges Bank locations where we see large aggregations of scallops. Tells us interesting information about where they like to live and what they like to do there. Shelf and near slope mixing where we see these different water masses along the area confronting different geographic boundaries. These allow us to sort of see things like expected gradients in DIC and oxygen as you move from the coast towards the shelf, where you're having sort of coastally indexed water meet with water that's usually older in age and has an aggregation of carbon and other features. And so we have ideas. We're not ready to declare anything about it, but ideas about potential upwelling zones of deeper water onto the slope. And we're interested in sort of seeing how that overlies the biological data. And do you see patterns in areas of abnormally high DIC near the shelf slope overlap with areas of low to disappointing scallop settling? You can see similar sorts of trends in our chemistry here. DIC and salinities, we're relatively confident that the trends we're looking at are evidence of conservative mixing, nothing out of the ordinary, and interested in sort of getting to more finer grained details of things like biological data, like the differentiation of photosynthesizers and their effect on the carbon and oxygen cycles in situ. So as a final note, summary of next steps, we have a number of sensors we're working on. Chanos 2 is just one of them. We have other low-cost pH sensors that we're looking at as sort of ship of opportunity or low-cost interested party sensors that are capable of doing pH measurements. So those are not tools dual parameter sensors, but a range of options here that are all out in the water currently doing sort of initial testing. And as we said at the top, Alec couldn't be here today, but I'm sure he and I would both welcome anyone who's interested in reaching out for any questions or ideas or thoughts. And with that, I'll pass it off to you.
Thank you so much, Evan. I have questions, so I will be emailing and following up. That was such an interesting talk. I'm going to hand to Carly, actually, because we're at the very end of our session.
Thanks, Molly. Let me just share. Awesome. All right. Well, thank you all so much. We'd like to thank our organizers, our speakers, Molly for moderating. This has been such a wonderful session, highlighting all of these exciting technology developments in ocean acidification. We'd also like to thank you, our audience, for joining us for Ocean Acidification Week 2025, not only in this session, but across all of the sessions we've had this week. We already have most of the sessions from the week up on the GOA-ON YouTube channel. So if you missed anything this week, I'd recommend checking out the playlist. We have a lot of great sessions and research from around the world being highlighted. If you're not already a GOA-ON member, we'd also love to have you join us. You can sign up on the GOA-ON website under the Members tab. And again, last but not least, just thank you all for making this such an exciting and invigorating Ocean Acidification Week. We look forward to the next time that we'll be seeing you all, whether that's virtually or in person. And yeah, thank you so much and enjoy your weekend.
Thank you.
Thank you.